Now,
Now, how come we could understand that the rough endoplasmic reticulum is the site of synthesis of proteins? This is by means of one of the technique is known as autoradiography. Here in the case of autoradiography, tissue sessions were prepared and definitely that should have radioactive isotopes. The preparation in this case maybe by means of incubating the tissue sections in a solution having radio labeled amino acids. So that radio labeled amino acids may be taken up by the tissue, and during the translation that radio labeled amino acids may be incorporated into the newly synthesizing proteins. So during the first few moments, we can see that the newly synthesized protein regions having radioactivity that should be only at the regions of their own synthesis. That means we are suspecting here that the rough endoplasmic reticulum is the cytosynthesis of. So during the first few moments, we could expect that the rough endoplasmic reticulum may be having the radioactivity. So now we have prepared the tissue sessions, and then thereafter we have to cover that tissue session with a thin layer of photographic emulsion. Photographic emulsion, the main component of it is silver ions. During the radioactivity, these silver ions will be converted to silver metals or metal silver. That is quite insoluble. After that, we can wash out the other soluble silver ions. See, uh, that means we can expect the silver metals only at the regions where just underneath of that the radioactive elements are present. So here these white regions are silver ions, and these dark regions are silver ions after exposing the radioactivity. That means they may be converted to silver metals. So after washing, those silver ions will be washed out, and the silver metals are only present here on the tissue surfaces. So these tissues then we can observe them under electron microscope. Then we will get an image of autoradiography.
Now, in an experiment using autoradiography, we can get to know the sites where secretory protein synthesis. For that, you have to get the pancreatic tissues, and that pancreatic tissues were to be incubated in a solution having radioactive amino acids. For a brief period of incubation in that solution, that solution means in that solution having the labeled radio labeled amino acid. These radio labeled amino acids were absorbed by the tissue, pancreatic tissues, and by the moment you have to take out the tissue and get a session, and that need to be fixed, and by means of autoradiography you can watch the real site of synthesis of the secretory proteins. The result is that this endoplasmic reticulum, especially the rough endoplasmic reticulum, is the site of synthesis of protein. Protein means these secretory proteins. So in this experiment, you have to expose the tissue for a brief period in the solution having radio-labeled amino acids. If you are exposing the tissue for a longer period of time, definitely then the labeled proteins that may be synthesized in the endoplasmic reticulum, that may start to move into the next compartment, that may be a vesicle, and from that vesicle to the Golgi complex, and from that Golgi complex to the next vesicle, and finally that may be secreted into the extracellular region. So all of these paths may be marked. That means if you are exposing the tissue for a longer period of time, then definitely the cytosynthesis of these proteins cannot be get determined. Now, to get to understand the site of origin of these kinds of protein and site of the first discharge, and then after that the second discharge site, and so on in a sequential way, you have to modify that experiment in a different way. Now this experiment is known as pulse chase experiment. So here in this pulse chase experiment, pulse means a brief incubation period, and chase means it is a period when tissue exposed to unlabeled medium. So here we have two solutions. The first solution is having labeled amino acids. Labeled means radio labeled amino acids, and the second solution is having unlabeled amino acids. That means it doesn't have any labeled amino acids. So here what we have to do is that at first you have to get the tissue. Here it is the pancreatic tissue, and you have to incubate that pancreatic tissue for a brief period of time in first solution. In first solution means that is a solution that is having only labeled amino acids, and after a brief period of time you have to take the tissue and wash it off to remove the excess isotopes present on the surface of it, and thereafter you have to put that tissue in the second solution. Second solution means that is having only unlabeled amino acids. That means it doesn't have any labeled or radio labeled amino acids. Then after that you have to take the tissue for particular periods of time, let's say at first minute or fifth minute, sixth minute, seventh minute, 8 minutes, and so on. So that means you have to get the tissue at regular time intervals, and then you have to prepare the sessions from that, and those sessions need to be auto-redigresively analyzed. And during that analysis we could get to understand that from the first session we could get to understand the site of synthesis of the protein, and the second and the subsequent sessions we could get to understand that where these radio labeled portion of the protein moves. Now the techniques have been advanced much more. Instead of more complicated autoradiography techniques, we can now rely on GFP proteins. GFP means green fluorescent protein. Actually, this is a kind of fluorescent protein that may be obtained from Aquaria victoria. Aquaria victoria is a kind of jellyfish, and the discovery may be felicitated with Nobel Prize in Chemistry. So this GFP protein's structural gene part may be incorporated into the gene of interest. So along with the expression of that gene of interest, the GFP protein also gets expressed, and then using fluorescent techniques we can track the cytosynthesis and the movement of the or trafficking of the proteins along the various cell organelles or along the various endomembrane compartments.